Juventas Therapeutics is going to present to us next, and the presentation will be given by Dr. Rahul Aras. Now, I'm not really that familiar with Juventas, so I'm interested to learn more about what this company is doing. However, reading through the perspective, reading through the company description, it's clear to me that the company is working on homing an SDF1. So if you have a massive infarct, you go into the emergency room, you're revascularized. There is a period of time at which the myocytes in the peri-infarct zone around the heart become overworked. They call out for help. They secrete HIF, or hypoxic-induced factor. Hypoxic-induced factor is transcribed into other growth factors like SDF1 or stromal-derived factor 1. So one of the key questions beyond just an acute ischemic event is how do you create a homing signal by which cell therapy can find its target? And my understanding is that's a little bit about what this company is about. So I'm looking forward to learning more. Thank you so much for joining us.
Thanks, Jason. So I appreciate the opportunity to present to you. Jason did a nice job giving the scientific overview. We are a venture-backed clinical stage company really focused on factors to treat cardiovascular disease. To his point, rather than deliver stem cells, what we've decided to do is look at the molecular drivers that either activate or enhance natural endogenous stem cell-based repair. What I hope to tell you about today is the biology behind that and how it's led to 2 ongoing phase 2 clinical trials right now. As mentioned, our lead product is SDF1, and the company is really based on discoveries by our scientific founder, Mark Penn, from about a decade ago, where he observed that if you, immediately after a heart attack, SDF1 is upregulated for a short period of time. The SDF1 works to repair the heart through recruitment of your natural stem cell pool. Unfortunately, that signal is short-lived and not sufficient for us to be able to see any significant repair. What he showed back then and what we've now done as a company is that if we can reestablish that signal at a time remote from injury, we basically trick the body into thinking it was just injured and activate your natural stem cell-based repair pathways. The nice thing for us as a company, this morning we talked a lot about mechanisms, is SDF1's really turned into a field. There are over 100 publications from about 20 different labs throughout the world, all looking at the mechanisms through SDF1 works. How SDF1 works. We know that it's home stem cells. We know they're bone marrow derived as well as tissue specific. We also know that leads to new blood vessel growth that's sustained for several months after our drug signal is gone. And lastly, we know that there's key trophic or direct effects of SDF1 in that it provides anti-apoptotic or anti-cell death signals to the tissue in which it's delivered. The way we're delivering SDF1 is through JVS100, which is a non-viral DNA plasmid. We know that it provides therapeutic expression of SDF1 for about 17 days at the target tissue, which is the optimal time for us to be able to see the kind of benefits that we want. It's directly delivered to the organ, and I think in a value-driven perspective, what we've done is created a very highly scalable, cost-effective, stable therapy that takes the process of cell therapy and really puts it back into a vial, which is something that, as we talk to potential partners and investors, gets back to the pharma biotech-friendly environment that they're used to. As mentioned, we are focused on cardiovascular disease. Like many of the companies in here, the biology is preserved and consistent through organ systems, but we took a very focused approach, assuming that we would first target heart failure and we could expand to other areas as we evolve. I think the panel this morning did a very nice job describing the needs in heart failure. It is one of the leading causes of death in the United States and throughout the world. We're specifically targeting symptomatic ischemic heart failure populations. So it is the population that Dr. Miller referred to as having high levels of hospitalizations and high mortality over a 1 to 2 year period. The other area where we're focused is in critical limb ischemia. So this is late-stage peripheral vascular disease. Again, relatively large population, about 3 million patients just in the U.S. I can tell you the outcomes of this are pretty morbid. About 25% of patients die within a year of diagnosis. 35% have a major amputation, and the cost of the healthcare system is very significant. What I'd like to do is start by sharing some of our phase 1 data from our heart failure study. This was actually just published last month in Circulation Research. It was a 17-patient open-label dose escalation study in what are classically Class 3 New York Heart Association patients. We tested 3 escalating doses, 5, 15, or 30 milligrams, delivered via an endoventricular catheter. We met our primary safety endpoint. That obviously was the key focus of this trial. I think interestingly, after 18 months, 15 of the 17 patients were still alive on a follow-up, which was probably greater than we expected if there was a placebo-controlled trial. We also saw very consistent improvements in quality of life, 6-minute walk, BMP, stabilization of volumes in EF, as well as about half the patients improving their New York Heart Association class. I think what was most intriguing and comforting about this data is really the consistency. We saw a clear dose response between the low and high doses that was really preserved in every single efficacy parameter we looked at. Based on that, we've now driven towards the initiation of a phase 2 trial. This again is going to be in ischemic symptomatic patients. We're enrolling it at 16 patients, 16 centers throughout the U.S. It's going to be 90 patients with 60 treated at 2 different doses and 1 control patient population. We're about halfway done with that. We'll obviously be looking at 6-minute walk, quality of life, BMP, but have also sized the trial, so we should be able to get preliminary data on hospitalization, mortality, and morbidity. The other area that a number of people in the field have migrated to, and for which there's good basis for efficacy coming from some of the autologous cell therapy trials, is CLI. And if you appreciate the mechanism through which SDF1 works, the benefits that have been seen in some of these other autologous trials suggest SDF1 should similarly provide some benefit. We're simply getting the cells to that same target region in a more efficient way. We have started a 48, or we're actually nearly complete with a 48-patient CLI trial. It's in Rutherford Class 4 and 5 patients. We're enrolling actually in 7 centers, 5 in the U.S., 2 in India. The distribution of patients is about even between the two continents. We are obviously looking at key parameters like amputation, wound closure, and cessation of pain medications, as well as surrogate markers like ABI, TBI, and TCPO2. We anticipate that data from this trial will start rolling out sometime in early summer or midsummer, with data from both trials really coming out around the turn of the year. As far as a company, we've maintained a relatively small team. We're up to 12 people right now. We've really focused our energies and our funding on clinical development. We've raised $30 million today. I think we're one of the few private companies in the panel, so I thought I'd just message to you guys that venture funding is still alive and available within this industry. We have a great syndicate behind that. As we look to the future, we're looking to start phase 2B trials in CLI next year and a heart failure phase 3, probably at the beginning of 2015. We envision that on development we can get to 2 BLAs within the next 5 years with between $125 to $150 million. So just to summarize, we believe, a unique strategy in the regenerative medicine field by using a factor that initiates the endogenous stem cell-based pathways. We have preliminary and safety and efficacy for our lead product, JVS-100. We do have 2 ongoing phase 2 clinical trials with data starting to roll out this July, and we look forward to sharing those with you at upcoming meetings. Some time for questions.
I know that we're running late, but I'd like to try to ask at least 1 or 2 questions. I see Dr. Miller sitting right in front of me. After the cardiology panel, Dr. Miller and I were talking about the fact that what percentage of congestive heart failure is not ischemic in origin, meaning that it's kind of everywhere in the heart. And so here's my question to you. When we think about using SDF1 or kind of creating a homing signal, it seems like it makes sense to create a homing signal in people that have, a long-going chronic nature of a disease. But is anyone home to receive that signal? In other words, the fact that they're in a chronic state, you're trying to recruit repair cells that can respond to that signal, whether they're CD34, CXCR4, through the SDF1 receptor, but are those cells present, or does it make more sense maybe to introduce an allogeneic therapy in combination with, say, an SDF1 delivered vehicle to the target ischemic organ and then allow that allogeneic therapy to be delivered? I mean, that combination seems like it makes great sense.
Sure. So I can answer that probably a couple of different ways if I understood the question properly. We all have nascent bone marrow-derived stem cells and tissue-specific cells that are at the damaged organ. Extensive studies in preclinical models have really shown that they home nicely to an SDF1 signal in a chronic setting, and they help revascularize the border zone, and that revascularization has a large role in improved function within those groups. As far as our ability to combine with other cells, that's possible. I think one of the, and to Dr. Penn's credit, who's the founder of this company, he took a very cell-agnostic approach to this field and has experimented with SDF1 plus cells and other models. But in our view, the SDF1 alone seems to be providing a significant benefit. We're seeing sustained angiogenesis for months after the SDF1 signal is gone, and we believe it provides a very cost-effective way removing the complexity of the cell therapy. Now, you can ask Dr. Miller and others sometime down the future, there may be room to combine these therapies, but right now we think that we have a viable therapy on its own.
Right. And one last question, which is in the CLI space. I know that Astrum will be presenting soon, and Astrum recently made a very key decision not to go after CLI. Very difficult to enroll, and it's very hard to find the sweet spot, whether it's Rutherford 4 or Rutherford 5 class, where you can really get patients that are far enough along that the placebo group will progress to, let's say, an amputation. We hope that doesn't happen. But in a clinical trial, given the complexity of CLI, how do you hope to tease out a signal in a small phase 2 trial that will have real value so that you can capture the investment dollars needed to go pivotal?
No, I appreciate the question. And I can't speak to Astrum; they can speak to their own decisions. But from our perspective, we've been targeting Rutherford class 4 and 5 patients. We've actually seen enrollment go quite nicely. This trial will be fully enrolled at just 7 centers in less than a year. We have seen anecdotally, and we haven't unblinded the data, some very nice stories about patients coming completely off pain meds, wounds closing, and obviously we don't know about amputations right now. But I can tell you when we talk to the vascular surgeons, one of the things that they're very intrigued by is, first of all, we're an office procedure. They literally open a vial, it's a 5-minute injection, and the patient can leave. So the complexity around the cell therapy field is removed. Also, the ability for these Rutherford 4 patients to come off their pain meds is significant. We have at least a handful of patients who have now gone back to work, able to get a full night's sleep. The ability to keep these people from moving into Rutherford class 5 is significant. Ultimately, one of the things they're most interested in doing is actually combining therapies such as ours and such as the ones being developed here with the procedures that they're doing. So they'll open a vessel, but then allow for the creation of microvascular growth through the delivery of those. We believe that their CLI itself is a very targetable and significant population, but within the PVD space itself, there's significant room to grow. And as far as the endpoints go, amputation is a key one, but there's several keys that will help. Just to expand on that one more step, one of the nice things about SDF1, if you look at our cost structure, the cost of goods for our highest doses could be about $50 to $100. Okay, so it's a completely different paradigm relative to an autologous cell therapy. We have a patient who enrolled in our trial, Rutherford class 5. 3 months later, they believe they got therapy. We hope we gave them therapy, otherwise it was really good saline. But their wounds closed, they're off their pain meds, they went from walking a couple hundred meters to about 1.5 miles. They wanted to get redosed. Now, clearly we can't do that in a clinical trial, but you can imagine a therapy that now you're providing a repeat dose at 4 to 6 months to a Rutherford class 3 patient to help eliminate the claudication pain. And because of the cost structure, the ease of delivery, I think we'll have this opportunity to walk backwards with the patient over time.
So walking backwards, I should share with you my ankle story from this morning. Thank you very much.
Thank you.